Hi, I'm Rick Amasino from the University of Wisconsin, and I'm going to give two talks about how plants know when to flower. First, I want to start with an obvious point. That's plants are stuck where they grow. And when plants are growing in temperate climates with seasonal changes, it's an adaptation for plants to be in tune with the seasons and be able to measure seasonal change. In many ecosystems, seasonal cues include changes in day length and changes in temperature. And in this part one, I want to focus on changes in day length and how it relates to plant flowering. This is a process called photoperiodism. Photoperiodism isn't unique to plants. Many animals have photoperiodic responses as well, such as certain birds migrating when day lengths change, or other animals hibernating. In a second talk in part two, I'll talk about another seasonal cue, changes in temperature. But first, I want to give a little background to the discovery of photoperiodism in plants. And it starts in a field in Maryland around the turn of the last century. So in the early 1900s, two scientists, Garner and Allard, found a huge tobacco plant in a tobacco field that wouldn't flower. It was a spontaneous mutation. Shown here is an example of that plant that we recently grew in a greenhouse with two of my colleagues, two graduate students. You can see that when this plant can reach huge heights, and it does so when it's grown in long days. Shown next to it is a normal tobacco plant that flowers at about the height of a person. So this mutant was called Maryland mammoth, and Gardner and Allard were curious about Maryland mammoth and wondered why it didn't flower. So before the frost killed it, they dug it up and brought it into the greenhouse, and it turned out it flowered during the winter. And they wondered why it flowered during the winter. They had many hypotheses as to what was going on to cause winter flowering in the greenhouse. For example, they wondered if growing in pots caused it to flower, or the light was more dim in the greenhouse, or the temperatures were colder. And they tested all of these hypotheses, but none of them proved correct as to why the plant was flowering. Then they had one more hypothesis that turned out to be correct, and that was it flowered in response to changing day length. And they showed this by building a house in which they could put the plants during the summer towards the middle of the day or towards the end of the day into this light-tight house, which they nicknamed the doghouse, and they could mimic the very short days of winter in the middle of the summer, and the plants would flower then as well. So they went on to also characterize photoperiod responses in many plants. And they found that some plants flower as the days were getting longer, others flowered when the days were getting shorter, and there were even more complex types of behaviors, such as plants that required short days, then long days to flower, or long days, then short days. But I wanted to show you just a few of the types of response types, the simple short day or long day responses. So shown here is a graph with day length on the one axis and flowering on the other. And if you look at a typical short day plant, when the days are long, for example 24 hours or getting shorter, they won't flower, but at a certain point they reach a critical day length. For example, 14 hours in this illustration. And then the plants, as the days get shorter, rapidly flower. Long day plants have a sort of opposite behavior. When grown in short days, they don't flower, but as the days get longer, they have a flowering response. Now, it's not just whether the day is short or long, but what the plant is really sensing is the changing of day length. And a good illustration of that is shown in a different response than flowering, but the induction of dormancy in a tree, an type of aspen that grows in the Arctic Circle. Here the plants don't begin to enter dormancy when there's continuous light of the Arctic, 24 hours of light. But when the day length shortens to just 22 hours of light, these plants are sensing this a short day. It's a short day response because they're sensing the days are getting shorter. Clearly 22 hours of light is a very long day, but this is a short day response because as days shorten, the response occurs. I wanted to talk about flowering itself. Shown here is a section through the growing tip of a plant. That growing tip is composed of stem cells called a meristem. And those are the cells that give rise to all the other parts of the shoot below, the stem and the leaves. And as you know, when a plant begins its growth, it's forming leaves and stem. But at some point, meristems can convert to forming flowers. Shown on the right is a plant that has undergone that transition, or a cross-section of it. Whereas you can see the plant on the right is making leaves. These other structures are cross-sections through flowers. And what I'm going to talk about is how that transition occurs. Now, this is occurring in the growing tip of the plant, but a classic experiment first done in the 1930s showed that it was actually leaves that were sensing the day length and sending a signal to that growing tip, the meristem, to cause flowering. And so shown here is an illustration of that type of experiment. These are plants of morning glory, which are short day plants. They require a long period of night and a short day in order to flower. These plants were both grown in long days, conditions where they normally won't flower, and the plant on the left is not flowering. But the plant on the right simply had a leaf covered with tin foil so that only the leaf experienced the short day, and the plant went on to flower. And in fact, other classic experiments shows that that signal moves from the leaves out into the meristem rather quickly. You can take a plant like morning glory and give it that long night that follows the short day, and then at the end of that long night, remove the leaves. And so what's shown here with the red graph is how long of a dark period is required if the leaves are removed. And it's about 12 hours of night. But if the leaves are removed at the end of the dark period, it only requires an extra 3 hours. At 15 hours, you can see flowering is occurring. And what that means is once there's commitment to flowering, after 12 hours of dark, it takes only another 3 hours for some sort of signal in this particular experiment to exit the leaves and start to travel to the meristem to cause flowering. There are other classic experiments. This one was done by a scientist named Jan Zevaert, and I want to show another one of Jan Zevaert's classic experiments, and that's to transmit this signal by grafting. Shown here is a leaf from a plant that's been induced to flower, grafted onto another plant that was grown in a non-inductive photoperiod, in other words, a day length that was not conducive to flowering. And these plants are flowering. You can see the small white flowers in the shoots that grow out under the influence of that leaf. Shown on the right is a leaf from a plant that was not flowering, and it does not have the ability to cause those shoots that grow out to flower. So clearly leaves are making a signal, causing the plant to flower. To discuss the concept of how plants flower in response to day length, I want to introduce the concept of circadian rhythms. Circadian rhythms are those 24-hour rhythms that animals, including humans, have. Also plants, fungi, and many microbes have circadian rhythms. If you've ever traveled to another part of the planet and found that you were awake in the middle of the night, that's because your circadian rhythm is set to where you came from, not to your destination. A hallmark of circadian rhythms is that they'll continue in the absence of the day-night cues that typically set those rhythms. And I'm going to show you an illustration of circadian rhythms in plants. This is a particular bean plant that's been kept in 12 hours of light, 12 hours of dark. So it has a particular circadian rhythm established. But the video will show you what happens if the plant is moved into continuous light. It still maintains a leaf movement, which is a circadian movement in plants. And this is from Roger Hangarter at Indiana University, who has a wonderful website called Plants in Motion that has many illustrations of plants and wonderful videos of the types of movements, including circadian rhythms, that plants undergo. So I want to play this movie for you, and you'll see this is just one example of a circadian rhythm in plants, that of leaf movement. What I want to focus on is how the circadian clock interfaces with photoperiod in plants. I mentioned earlier morning glory. It's a plant that flowers when the days are short and the nights are long. What I also want to mention is it flowers in response to a single long night or short day. And in plants like this, they will flower to that long night, even if that night is incredibly long, for example, on the order of 60 or more hours. And so this leads to a classic type of experiment that was done that showed that interface of the circadian rhythm and photoperiod sensing. So that experiment is illustrated in this slide. And what the experiment that this represents is, first of all, the plants were grown in light. See the bottom bar that shows the light period. But the dark bar is the transition to a very long night that's on the order of 60-some hours. And what's done in this experiment is everywhere you see a data point, that a flash of light is given that would interrupt the flowering of the plant, because these plants that respond to a single dark period, or short day plants in general, are incredibly sensitive to a flash of light interrupting the ability to flower. But when you have this long night, what was measured are flashes of light given at various times, shown by the data points, and asking whether it was effective at interrupting flowering or not. And you can see that at certain points, the flash of light really has no effect, that there's very good flowering, for example, when the light is given at certain times. At other times, flowering is completely prevented by a flash of light. The red line simply shows a smoothed out opposite plot to what's shown in the black lines, which is flowering. But the key point is this. That is that there's a circadian rhythmicity to the ability of light to interrupt flowering. And that shows this linkage of circadian rhythms and flowering. And it's this linkage that led to the idea many decades ago by a scientist named Bunning of a coincidence model for how photoperiodism works in plants. And that coincidence model is that there's some underlying circadian process that's the same in short days and long days. And that's shown here in yellow. But that in long days or short days, there's different amount of overlap with day length, or with the day, or the night, with that underlying circadian process. So shown here in long days, for example, you can see there's an overlap of light with that underlying circadian process, which could be, for example, the appearance of a transcription factor or another type of molecule. In short days, there's not that overlap. And that overlap or lack of it could either lead to the promotion of flowering or the inhibition of flowering. And it turns out that work from Arabidopsis genetics, as well as rice genetics, gave us the key clues to understanding the molecular details of this coincidence model. So I want to give you the take-home message and then talk about some of the data that has led to this model. So here's a part of that overlapping coincidence model. And key to flowering in plants is that there's a coincidence of the accumulation of a particular protein called CONSTANS. And it regulates the expression of that flowering signal that I mentioned earlier that moves from the leaves to the meristem to trigger flowering. And by the way, that signal is called Florigen for its ability to cause flowers. And it's also encoded by a gene that's called FT. So I may speak of Florigen or FT because we now know the identity of Florigen from some experiments that I'll describe. First, I want to mention the ability of genetics to help sort out some of these molecular mysteries that have been around for a while. And so much progress in understanding how flowering works at a molecular level came from advances of genetics in both Arabidopsis and in rice. Shown here are some Arabidopsis mutants, but I want to start with showing the response of Arabidopsis to photoperiod. Shown here are Arabidopsis wild type in both long days, and long days trigger flowering. I've mentioned short day plants before. Arabidopsis is a long day plant, so it flowers rapidly in the long days. And also a wild type in short days, where you can see flowering is delayed. And so I also want to note that there are two classes of photoperiod mutants that one can find. So shown with the red arrows is one class. In long days, the mutant GI, or for gigantea, is a mutant that's blind to photoperiod. It behaves, as you can see, like wild type in short days. In other words, this mutant no longer senses the long days. There are examples of mutants, though, that in short days are sensing long days. Here's an example here of the mutant early flowering 3. Note that in short days, it's behaving as the wild type in long days. So we can find mutants that where the ability to sense photoperiod is blocked, or whether it's activated regardless of the day length. And then cloning the genes involved in this process and asking what they do at a molecular level has led to an understanding of how photoperiodism works in plants. A key observation was that the expression of the gene CONSTANS was under circadian control. This is work from George Coupland and his colleagues. And shown here in the top panel is looking at the RNA levels of the gene CONSTANS throughout a 24-hour cycle. And you can actually see that it varies depending upon the time of day. It's under circadian control. And as I said, Arabidopsis is a long day plant. It flowers in long days because you can see by the boxes that during a long day, which is illustrated, the period of light here is shown by the white bar, and black corresponds to when the night is occurring in this experiment. What you can see is there's some constants expression towards the end of a long day, and if that constants expression overlaps with light, then the Florigen gene, FT, is activated, and that triggers flowering. But it's actually a little more complicated and sophisticated than this simple system, because Coupland and his colleagues also showed that constants expression was also controlled at the level of protein stability. Here again is that image showing the RNA levels fluctuating in a circadian manner throughout the day. But shown at the bottom is that one can get constant levels of expression of the constants gene if it's driven by a constitutive promoter. But the interesting thing is, even though it's driven by a constitutive promoter, in this case the 35S promoter, which is from a plant virus, the levels of protein shown here are only, protein is only accumulating during the light, despite the constant expression of the gene. So there's obviously another level of control just in the level of transcription and RNA accumulation. And a number of labs have worked out what's occurring here, including the lab of Takato Imazumi at University of Washington. So shown here again is a light-dark pattern, starting with dark, transitioning to light, and then to dark again. And it turns out that there's a protein degradation machine that's always trying to degrade the constants protein. It's particularly effective at degrading the constants protein in the dark. But in the light, there's other plant light receptors, such as the FKF protein, which absorbs blue light, associates with GI, which is the gene encoded by one of the mutants I've showed earlier, gigantia. And this protein complex actually binds to the constants protein and protects it from the degradation machinery. So there's various levels of sophistication of this photoperiod system control. And overall then what's happening is in the leaf, when the days are long for an Arabidopsis plant, Florigen is produced. It travels up to the meristem. And in the meristem, it partners with another protein known as FD. And the partnership, a dimer of these two proteins, triggers a cascade of events that lead to flowers being formed in the meristem. And I wanted to also say a few things about the movement of Florigen and its partnership with FD. There were some classic experiments that gave a hint that FT was likely to be moving from leaves to the meristem and to join this FD protein to together promote flowering. One of the bits of evidence was that FD mutants suppress the rapid flowering of FT overexpression. One can take Florigen and cause it to be expressed at high levels, and plants flower very rapidly. But in screens for mutants that suppress that rapid flowering effect, a gene called FD was found to be a suppressor. And also, at a level of protein interactions, the FT protein was found to interact with FD, both in assays that are conducted in yeast as well as in vitro. But the key thing was that FT is expressed in the vasculature of leaves, but FD is expressed in the meristem. So that led to the hypothesis that FT must be moving from leaves to the meristem and could be Florigen. And that was finally shown by many groups who actually labeled the FT protein with a tag that could be visualized. And in innovative experiments like shown here, would graft a part of a plant, shown here in green, the top part, the shoot, that could express this labeled Florigen protein to a part of a plant, this rootstock, that was lacking the gene. The gene had suffered a mutation and it wasn't expressed. As you might be able to see in some of these fluorescent images, one could detect the movement of the protein from the shoot down into the roots, clearly showing that this protein can move throughout the plant, much like hormone proteins can move in animals as well. Now, you might notice this, that we're showing movement in this particular experiment that I've used to illustrate from the shoot to the root. Yet in flowering, movement is occurring to the meristem as well. And that's an opportunity for me to introduce the idea that this expression of FT under the control of photoperiod can control many processes in different plants, not just flowering. And some of those do require movement from the leaves to the root system. And a good example of that is shown in the next slide. That is, in potato, the ability to form tubers, those edible parts, is often under photoperiod control. And it turns out that expression of FT is responsible for that ability of potatoes to sense photoperiod and form the tubers that we eat. And that's a function of FT moving from leaves down into the root system. Also, in other studies, and a reference is shown here, that the ability of constants to regulate when FT is expressed also can control when plants enter dormancy. So in the fall, changes in day length that trigger plants to enter a dormant phase to prepare for winter can also be controlled by this process. So this is a very ancient system controlling many photoperiod responses in plants. Now finally, what I wanted to mention is the development of flowers themselves. So shown here is a group of Arabidopsis flowers. The meristem forms an inflorescence of many flowers that undergoes continuous growth and formation of flowers. And what's occurring when FT or Florigen moves to the meristem, partners with FD, is it sets off a cascade of expression of master control genes that cause those cells in the meristem not to form leaves any longer, but to shift their developmental program to form flowers. And here are just some examples of the rapidity of that response. For example, when Arabidopsis plants are shifted from short days to long days at time 0, you can see that very rapidly, within 18 hours, you can see changes in gene expression at the meristem. These are sections through a meristem. The color is a result of hybridizing a probe that can fluoresce to certain genes as they're expressed. And you can see rapid induction of genes in meristems of plants after they start producing Florigen. Here's another example for the gene apetala. And so what we now understand is the molecular pathway by which photoperiodism occurs. And it's this cascade of gene expression from the signal that moves from the leaves to the meristem that causes this huge change in meristem development from a vegetative meristem shown on the right. Vegetative refers to the fact that the cells produced from this meristem form leaves, and it causes it to transition to those cells that the meristem's forming to differentiate into flowers. So I'd like to thank the National Science Foundation, the USDA, and also the National Institutes of Health for, over the years, funding our work on flowering research.